So it's a great pleasure to introduce Jesper Madsen. Jesper is a professor at the Department of Biochemistry and Molecular Biology at the University of Southern Denmark, and a member of the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease at the Broad Institute. I'm really excited for your talk today, Jesper, and Jesper plans to speak to us about interpreting clustering of single-cell transcriptomes.
So thank you again for inviting me to give this talk. I will try to talk to you about interpretable clustering of single-cell transcriptomes. It's some work we've been doing over the last year or so to try to develop a new algorithm for batch integration. And since this is a workshop, I just started very basic. Because there might be students here who don't know what all of these technologies we're talking about is about. So back in the day, for me at least, about, let's say, more than 10 years ago, when we wanted to study the transcriptomes of tissues, we isolated RNA from the entire tissue, and we did this bulk analysis where we took all of the RNA, we sequenced it, and that gave us a readout that was the average expression of all of the cells in these tissues, here shown with 5 or 6 different genes in one color, just stacked. And that did not allow us to unmask this heterogeneity of cell types. So then enter 2016, maybe around that, 10x Genomics commercialized single-cell genomics or single-cell transcriptomics. It had been around for a while in labs, developing the methodologies, but the commercialization of it really drove the adaptation in the field. And now a lot of labs around the world, including us and Broad, and certainly many labs are doing single-cell analysis. And here we are dispersing the tissue into single cells, a single nuclei, and we're then doing transcriptomics in each cell to measure the transcriptomes of each cell. This allows us to do many types of analysis, for example, reveal the heterogeneity and subpopulation expression. It of course has some caveats, which is what I will talk about today. We can use this data set, these types of data sets, for many different things. I like to think about them in sort of steps of advancedness. So the primary analysis, I would say, is to define cell types and states. This is also, for example, usable for these eQTLs or other types of QTLs we want to do within either distinct states or along continuous manifolds. Then we can do what we could call primary analysis. We can do differential expression analysis to compare different states or different cell types. We could also have some covariates in here that could be, for example, different types of donors, and then we could compare cells within cell types across donors. Similarly, we can do differential composition analysis, asking if the composition of the tissue is changing across case control studies, for example. Then we can also get into more advanced analysis that could be RNA velocity, where we are trying to use the information about exon and intron reads to measure the change in gene expression, or the predicted change in gene expression, and then relate the future state of cells to the present state of cells, thereby deriving a trajectory. Example here is showing development of the pancreas, where these endocrine progenitors, let me just get a point, endocrine progenitors differentiate towards the different, or at least 4 different endocrine lineages missing the gamma cells for some reason. And another type of advanced analysis or secondary analysis would be cell-cell communication, where we want to disentangle how cells communicate with each other. Some cells express receptors, some cells express ligands. They talk to each other. We want to find out how that network is working. Or it could be, as we have seen, multiple instances of during this workshop to integrate genetic information or other types of information. But in order to do that, we need to have a data set that is amenable to analysis. And what we often see is something that looks like this when we analyze our data sets naively. So here we have the same tissue, the same sample, but analyzed with two different technologies. Here is very unrelated technologies, well-based technologies, and two droplet-based technologies. And we see that the cells are separating by batch. And if we look at the cell type labels, we can see that the same cell types are actually also separated by batch. So for example, here we have B cells that are separated in these two batches. So in order to enable all of these more advanced analysis, we need to take care of these technical effects or nuisance factors, if you will. And the way we do this is that we use batch correction methods. And the aim of batch correction methods is something like this, where we want to sort of mix the cells from different batches. It can look like this or this. And what should then happen is that the cell type should now, or the cells should now separate by cell type rather than by batch. And that seems to work quite nicely. At least in these two examples. So there are different and already many different batch correction methods available out there. And in a recent benchmarking, the TICE group from Germany, they set out to benchmark these methods to find out which methods are good and which methods are bad. And what they did is that they took a bunch of different data sets, and then they integrated them with different methods, and then they evaluated how much batch effect could be removed. So in other words, how mixed are the batches after integration. And crucially, I think they also evaluated how much biological variance is conserved after batch correction. And they did that by different approaches, such as the conservation of highly variable genes before and after batch integration. The results, I think, are quite interesting. There are a few things to notice. So these are the top 20 results from that paper, and this is 5 data sets and 2 simulated data sets. And what we immediately saw was that the semi-supervised or supervised methods are the most powerful batch integration methods currently. But what we also saw is that the linear or the interpretable models generally underperform. So for example, down here we have LIGA, which is a linear non-negative matrix factorization based method, which doesn't actually perform very good compared to the nonlinear methods. I think although the authors here did evaluate the conservation of biological effects, I think they missed perhaps one crucial element of preserving biology, which is starting to appear more and more in the field, which is overcorrection. So here, the Schat group from the Icahn School of Medicine, they made an in silico mixture of single-cell RNA sequencing from the brain and from the homeostatic intestine in mice, and then they ran batch integration, and they then evaluated how dissimilar are the data sets after correction. And these two data sets, they shouldn't integrate. But what they actually found is that some of the methods integrate them quite substantially. Here is Surat and Harmony, and to some extent LIGA, that mixes these data sets even though they are distinct. And in another data set, Xiong and colleagues, they made an in silico mixture of 2 different liver data sets. Crucially, these liver data sets, they contain different subtypes of hepatocytes. Here, all of these cell types here are hepatocytes, but they are marked by different marker genes. They also have 2 different subtypes of NK cells. And when they ran batch integration, they saw that the methods tend to integrate or mix these hepatocytes or different states of hepatocyte, even though they should be separated. So that's sort of cell state erasure that can occur because we overcorrect these methods. Finally, a recent bioRxiv paper from the Theis group, again, this is from approximately 2 weeks ago. They also demonstrate overcorrection in deep learning-based integration methods. Here they integrate tissue foeva, tissue periphery, and organoids. And they see that Mueller cells marked by RHCG, they co-localize in space, but there's actually ample evidence in the field that Mueller cells from the foeva are transcriptionally distinct from Mueller cells from the tissue periphery and organoids. So batch correction can be a problem. It can overcorrect and it can erase cell state differences. So what we set out to do is that we wanted to design an algorithm for batch integration, which is interpretable, which is robust against overcorrection, and which has state-of-the-art performance. Our take on that is called jointly. And jointly works sort of like this. You have 2 different data sets that you want to integrate. Then you put it into jointly, and out you get an embedding, which you can use for joint clustering. And you also get gene modules that you can use for interpretation. And of course, jointly isn't really a flat box. It looks like this, schematically at least. It is a hybrid, nonlinear and linear, non-negative matrix factorization with this particular loss function. I haven't really derived all the math here, but I've included sort of an intuition of what each of these terms do. So the first term here in green is corresponding to this sort of visualization. And what we do here is that we do a nonlinear mapping of the data set using an alpha decay kernel that allows us to measure the distance between cells in nonlinear and local space. So we can ensure that our algorithm accurately reconstructs the nonlinear and local neighborhoods. Then in blue here, we have a graph regularization term. Here we're using the shared neighborhood graphs, or shared nearest neighborhoods. That is a more global distance metric. So here we are ensuring that not only with the green term are we preserving nonlinear and local distances, but we are also preserving global distances. And finally, we have a linear module, which is an interpretable module of genes, where we are minimizing the reconstruction error across data sets. So this term is actually the only that is shared across data sets, and all of the other terms are data set specific. And with this algorithm in hand, we set out to test it. And we started with a real, but it's kind of a toy data set, but it is a real data set. Here, Wegman and colleagues, they mixed 8 different cell lines and they performed single cell RNA sequencing on them. And it doesn't really have any batch effects. So what we did is that we simulated some batch effects in this. And it looks like this. So what we did is that we simulated a nonlinear and also a complex batch effect that doesn't affect every cell type equally. So you can notice, for example, that the HCT116 cells are fairly well mixed in the naive PCA space. But for example, the RAMA cells are quite significantly affected by batch effects. And if we try to cluster in this using the naive analysis, we can see that we can not actually recover the true cell types. Here we see that one of the clusters of RAMA cells is actually assigned to the UCAT cell clusters. And this shouldn't, of course, happen because RAMA cells are a type of B lymphocyte, whereas UCAT cells is a type of T lymphocytes. And I should stress that the way we did this is that we actually tried clustering at many different resolutions, and then we took the best one. So this is actually the best that could be achieved if we used just the naive analysis. Then if we apply jointly to this data set, we can see that suddenly we only have 8 distinct regions in our UMAP space. And these correspond actually perfectly to the cell types in the data set. An interesting feature, if you will, is that we can actually see that cell types with a high batch effect are sort of different, and that will perhaps be important later, that it seems that jointly is not forcing cells on top of each other, but it's minimizing the distance between related cells. And that's actually quite substantial in reducing overcorrection. So we're quite certain that in this data set, at least, that jointly can recover the original data sets or cell types with high precision. What we then wanted to just do is that we wanted to evaluate these interpretable factors in the same data set. And we took one factor here is a factor. When we score that factor across the cells, we can see that it is highly scoring specifically in A549 cells, which is a basal epithelial cell from the lung. And we can also see that the genes associated to this factor are genes related to epithelial cells or epithelial cell identity. And we can see if we look across additional factors that jointly is actually discovering gene programs that are highly cell type specific. Yeah, and if we look at these factors and compare them to each other, that's what we did here. So on the x-axis and on the y-axis, you have the same factors, and then we just correlating the scores. We can see that the scores are generally not strongly correlated to each other. And the same is true in the second batch. And we can also see it across batches, indicating that the gene programs identified by jointly are orthogonal to each other. One thing we had available to us in this data set is that we also had the data available without batch effects. So we actually also ran jointly on the same data set without batch effects, and then compared the modules, the interpretable modules that we found. And we can see here along the x-axis here, we have the factors in the data set with batch effects, and on the y-axis, we have the factors again, or another set of factors derived from an analysis without batch effects. And we can see that the modules are highly similar. They're not ordered in the same fashion, but there's approximately one-to-one correspondence between the genes associated to factors with and without batch effects. So that led us to conclude that jointly can identify orthogonal gene programs, and it does so almost independently of batch effects. So with this in hand, we had an idea that jointly could be a pretty good batch integration method that would also be interpretable. So we set out to benchmark it. And in order to benchmark, you need to decide on a few different things. We needed to decide on who we wanted to compete against. And we also needed to decide on how we define what is true and what isn't. So in order to define the competition, we went back to this article, and we took the top 5 non-linear methods that performed the best from this study, and we excluded the supervised methods because jointly isn't supervised. And we also spiked in Leica here, which is similar to our method in the sense that it is NNF-based and interpretable. And finally, we also included a recent foundational model for single-cell RNA sequencing, scGPT, which we transfer learned to do batch correction and evaluated in the same framework. So that was selecting the competition. Then we also needed to set the ground truth. And the common way of doing this is to use published data sets and use the author labels as ground truth. Here's an example from the human liver, which is a data set that we wanted to use. But looking into the literature critically, we noticed this in the method section from this paper, that the authors in this case used one of the methods that we wanted to benchmark against to actually obtain this embedding and cluster. And in our opinion, that sort of means that there is a confounding effect by methodological choices by the original authors that could potentially favor contemporary methods. So we wanted to define a truth in a way that did not depend on batch integration. So what we noticed reading papers in this field, specifically from label transfer, is that actually support vector machines seems to be quite good at label transfer. So here's a article where again, the TIES group defined this SC poly model and evaluated its ability to do label transfer in different tasks. And what you can see is that actually the yellow bars are the highest, which is just an SVM. In another study, also from the TIES group, they evaluated how SC-ARTIST works, and they found again that SVM is actually outperforming these deep learning-based methods. And finally, in another study again from a different group that tested a lot of different methods, they actually also found that SVMs with a rejection option seems to be the most highly performant method for label transfer. So what we decided to do is that we decided to transfer labels using an SVM with a rejection option to relabel every data set that we used in our benchmarking such that the labels were not dependent on the batch correction methods used by the authors. And the way this looks is like this. This is an example data set from the human liver with author labels. And then these are transferred labels, and it looks quite similar. The resolution is a little different, and the naming convention is also a little different. So for example, down here, the authors call these cells app T cells, whereas the label transfer just calls them T cells and so on. But if we compare the labels, we can see that almost all of the, for example, almost all of the endothelial cells, or are called, sorry, the other way around, almost all of the LSEC1 cells are assigned to endothelial cells. So even though the labels are not the same, the purity is actually quite high, and approximately 90% of the cells were assigned to the most prevalent group. What that means is that approximately 90% of the cells were assigned to the group that most of the cells in that group was assigned to. So now we had the ground truth, and we also have the competing methods. So now it was just about running everything. And this is an example of what it looks like. Again, from the human liver. Here we have the methods that we included jointly: SCVI, SCGT, Harmony, FastMNN, Scanorama, Seurat, Leica, and then the naive analysis. And we did this in the same way as before. So we used the available vignettes from the different tools to decide on how many dimensions each method should use, so that it was derived from the author's suggestions. And then when we clustered it between 2 and 50 clusters, and for every method reported the best possible adjusted rand index, which is a measure of the agreement between clusters in this case and labels. And what we can see for this particular data set is that basically all of the methods, with the exception of Leica and PCA, perform similarly, achieving an RI at approximately 0.9. And they are also all tripped up by the same cells, which in this case are these cells down here where there are actually 3 different types of endothelial cells that are clustered together in one tight cluster. And we can't resolve that, and neither can any of the other methods. We expanded this to not only human liver, but to 5 different data sets. And we then summarized it in a rank. And what we found is that there's a few methods that generally perform the best. This SCVI, it's jointly, and it's harmony and fast dependent. And although SCVI is ranked 1 here, it's actually marginally higher than the other 3 methods. What we also did is that we measured what we call worst performance. So here, global performance means the ROI if we look at every data point in the data set. But what we also did is that we zoomed in on every single batch in each method, and then we evaluated in each batch how well is the data integrated. And then we report on the worst batch. And here we can actually see the picture shifting a little bit. So if we look at in the worst batch, we can see that now jointly SCVI and Scanorama is actually the best performing methods, whereas, for example, Harmony, which was very good overall, is actually quite poor in the worst data set, suggesting that both SCVI and jointly are capable of a balanced batch integration. So if we look at the 3 goals that we had, I would say that we are on par with state of the art, which is, I think, satisfactory compared to that we are working with a linear, or at least partly linear model that is interpretable. And we can see that compared to the other interpretable models down here, we are much, much, much better. So the next thing we wanted to evaluate was the robustness against overcorrection. The way we did this looks like this. So we took 6 donors from human liver. We took 12 donors from human islets. We mixed the data set, and we integrated across the 18 donors. So we didn't tell the algorithms that it was different tissues, and then we just asked the questions, how much do the 2 tissues mix? And also we asked how well is the cell types in each data set then batch corrected if we look at them in isolation. So the idea here is that this is a sort of an extreme way of recapitulating a data set where you have healthy individuals and sick individuals, where there might be cell state differences or even cell type differences. But the algorithms don't know that, and it's convoluted by intra-donor variation. The results look again like this. It's quite a busy slide, but I think it helps a little bit to have everything on the same slide. So what we have up here is all of the methods, and then we have the cells colored by donor. In green, orange, red colors, we have the pancreas samples, and in the purple to blue colors, we have the lung samples. And similarly, down here, we have the cells colored by cell type. And again, in orange to green colors, we have the pancreas. And in blue to purple colors, we have the lung. And immediately, we can start to notice a few things. If we start with the naive analysis with PCA, we see that there's quite good separation of tissues, as we would expect. But there's actually quite a poor integration of cell types. So for example, here we have alpha cells, which are also present up here from 2 different donors. That's not really surprising. SCGPT actually does a quite good job in this task. It doesn't really mix the tissues, especially, but it doesn't really have a lot of power to resolve individual cell types in, for example, the pancreas. Liger and Surat is really extensively overcorrecting in this case. We see that they are completely mixing the 2 tissues and placing totally unrelated cells on top of each other. Scanorama does a very good job. It actually nicely separates the two tissues, and it also has decent power to separate cells. Fast MNN also has a good separation between tissues, but it lacks the power to resolve individual cell types in each tissue. And finally, our method, SDVI and Harmony, are all doing a good job at separating the tissues and also seem to have a decent power to separate the individual cell types in each tissue and batch integrated. So since these three methods were really the one that had the best performance in the batch integration task, we zoomed in on those three a little bit, and the embeddings look like this. And what we noticed immediately was that there's a difference, especially in this cell type here. So these in blue here, we have endothelial cells from the lung, and in green here, we have endothelial cells from the pancreas. And we can see that jointly and SCVI are actually keeping those two mostly separated, whereas Harmony is integrating them on top of each other. So we looked closer into that, and what we noticed was that both types of endothelial cells are definitely bona fide endothelial cells. They have high expression of canonical endothelial cell markers, but they are actually also quite distinct. So there's a large group of marker genes that are specific for the lung endothelial cells, and also a large group of markers that are specific for the pancreas endothelial cells. And these markers are conserved across donors, indicating that it's not spurious expression of batch affected genes, but actually bona fide differentially expressed genes. And we could actually map that back to differences in immune signaling between the tissues, which is likely to occur between the lung and the pancreas. So it seems here that Harmony is also susceptible to cell state erasure, whereas jointly and SCVI are doing a good job at separating cell states between samples while integrating across donors. We can summarize all of this again down to a few ranks. So here we have ranks that indicate how well the methods integrate cell types and removes batch effects within each tissue, and then also how much it overcorrects, and we can then summarize that into an overall rank. And actually SCVI is getting a lower rank here, primarily because it is struggling a bit to resolve some of the pancreas data here in the middle. And so that was, of course, one comparison of data sets, and we extended that by making all possible combinations of the 5 data sets that we had in our benchmark. And what we can see is that jointly ranks second or first in 9 out of these 10 combinations. The only where we rank bad here is the liver and PBMC mixture, where we rank 5th. So in summary, I would say that jointly is overall pretty robust against overcorrection as well. Then the final thing to evaluate is this interpretation. What is the value of having an interpretable model? Does it bring anything, or should we just use these uninterpretable models that might have as good or perhaps better in the future integration performance? So in order to evaluate that, we looked at a data set from human adipose tissue from the Rosen lab, and we integrated it using jointly, and we labeled it and arrived at this particular UMAP. And we can see here that our UMAP, our marker genes are, our labels are well supported by established markers of these different cell types. What we then did is that we took the interpretable factors from jointly and we scored them for each of these cell types. And we saw a few different interesting things, I think. First of all, we saw some factors, such as factor 1, which is high across immune cells. But we also saw that factor 15, for example, is high across only myeloid immune cells, and factor 4 is specific for macrophages. So we see this multi-level expression or gene programs that we can dissect using jointly. We also saw a lot of cell type specific modules, and we also saw a few modules that actually did not appear to be expressed particularly highly in any cell type. And interestingly, we find that this factor 14 is actually enriched for genes that are likely to be ambient genes. And if we remove factor 14 before running the embedding and clustering, we actually see an improvement in the RRI of the analysis. So maybe we can also use the interpretability here to actually find relevant factors to use for clustering and embedding. Another thing we did is that we took the genes associated to each factor and we put them through gene set enrichment using a database of cell type markers. And we then ask if we can find the correct cell type just by gene set enrichment. And in many cases we can. So for example, factor 2 here is highly expressed in mesothelial cells. And we can also see that the most strongly enriched term here is mesothelial cells. Factor 5 is vascular smooth muscle cells, and you can see here is mural cells, and mural cells is a combination term for pericytes and vascular smooth muscle cells, so that's also correct, and so on. So we can by looking at the genes assigned to these factors, we can start to get an idea about the cell types in our data set, and also perhaps some of the technical factors that we can look at. We then also compared the modules to post hoc analysis, so to DE analysis, to marker gene analysis, which is the alternative to looking at the interpretable factors. And here we also see some interesting things. We see that the genes that are DE, so they are positive markers for one cell type, and are also in our interpretable modules, actually have a quite high AUC score, meaning that they are very discriminatory. They're very discriminatory for cell types. Whereas the DE genes, the marker genes, so they're significantly enriched in one population, but they're not within our interpretable modules. They are actually not at all discriminatory for cell types. We also see that the reverse is true. So genes that are not marker genes, but are interpretation genes, they actually have an AUC below 0.5, indicating that they are marking cell types by absence rather than enrichment. And of course, genes that are neither DE or interpretations are not providing any information about cell types. What we also did is that we performed this DE analysis across batches and then asked if the genes that are DE are conserved across batches. And here we see the same tendency that genes that are in our interpretable modules are also more consistently DE genes across batches than DE genes that aren't. So we find with the interpretable modules, we find more discriminatory genes, and we also find more consistent genes. If we look at the enrichment of the genes across the cell types, we see, again, a few interesting things. We see, for example, factor 13 here is very specific for adipocyte genes. But we also see that some of the factors, as I mentioned, are actually shared across multiple cell types. So for example, here, factor 10 is mostly in fats, but also in adipocytes. And fat is a fibroadipogenic progenesis, which is a precursor state for adipocytes. Or at least some of them are. And if we look at the enrichment of factor 10, we can see that it actually has a quite interesting enrichment. So it is these cells right here are adipocytes, and these are the FAPs. And we can see that factor 10 is high in a subset of FAPs and also in a subset of adipocytes. And if we look at the genes associated to this module, we can see that it's genes associated to insulin signaling. And if we score it by gender, we can actually see that these genes particularly are high expressed in females than males, which is consistent with knowledge that the insulin sensitivity of adipose tissue is higher in females than males. So not only can we find these highly consistent genes, but we can also find factors that are not conforming to this notion of having distinct cell types. So factor 10 here is not only not enriched in only one module or in one cell type, but it's also actually only enriched in subsets of cells within two different cell types. So that would have been difficult to identify using traditional DE analysis, looking for genes that are enriched in an entire cluster. Finally, for interpretation, we also looked at temporal signaling. So here we took a mouse gastriculation data set, and we looked specifically at this arm down here, which is a differentiation of red blood cells. So down here we have the mature red blood cells, and up here we have progenitor cells. This data set has 3 batches, and we integrated it jointly, and then we colored it by the same labels as the original authors here. And we can see that jointly is capturing this continuous space here, that we are ordering the cells in the correct order. And to support that even further, we also performed a SC-Velo analysis here to obtain pseudotime estimates. And we can see if we color the jointly embedding by pseudotime, that jointly here is capturing the continuous trajectory of this differentiation quite well. And if we then use our interpretable factors, we can see that the interpretable factors in this type of data set has a temporal profile. So we see a bunch of factors that are especially expressed high in early differentiation here. Then we have some that are temporary, and they're sitting right on the boundary of going from the progenitors to the first erythroid state, indicating that these might be involved in transition. Then we have some early and late mid active factors and some late factors. And gene set enrichment analysis of these different groups of factors reveals a rolling signaling cascade that is highly that has been at least in steps validated in the field. So insulin signaling is important in early differentiation, then KIT signaling, TGF-beta, and then finally there's of course heme biosynthesis, which is the hallmark of the red blood cell. So if we then go back to these goals that we had going into this, I would say that, in my opinion, interpretable batch integration definitely has some merits. We can find very good marker genes and also good gene programs that are relevant for the biology in the tissue. So I would also say that that can be important. So as a last thing, we wanted to just showcase how we can then use jointly to analyze real data sets and to build, for example, a tissue atlas. So what we did is that we took 6 different studies that had performed some type of single cell RNA sequencing in a fat tissue. So that could be a subcutaneous or a visceral adipose tissue, either analyzing the whole tissue or only the stromovascular fraction, which is the part of the tissue that is not adipocytes. Then for each of these studies, we used jointly to batch integrate and cluster, and then we labeled the data sets. So we had these 6 labeled data sets, and then we used a supervised integration method, particularly SC-ANVI, to obtain a latent space. And the reason for doing this is that is that jointly does not allow for adding in new data sets, so it's not an online algorithm. But SC-ANVI is. So doing it in this fashion allows us to, in the future, add a new data set into this existing latent space, or to contextualize new data sets. And we were quite happy about this latent space. I think we have seen some interesting things here. One of the interesting things, for example, is the presence of Schwann cells. Schwann cells have been reported to exist in the adipose tissue and have been seen using immunofluorescence, but the transcriptomes of the cells has actually never been measured, and they haven't been discovered in each of these studies individually. And we also see other interesting populations. For example, there's a foam-like subtype of macrophages, which appears to be tissue-specific or depot-specific. We're really confident about our labels. So I think we have really strong support for the labels that we generated. We see that for the broad categories here, that there's very distinct expression of marker genes for a bunch of known marker genes, and we can really zoom in. For example, if we zoom in here on the vascular endothelial cells, we can separate the vascular endothelial cells into venous, capillary, and arterial vascular endothelial cells, and they have really clear and distinct markers that are consistent with data in the field. The same is true if we look at the mural cells. We can separate those into smooth muscle cells and pericytes. We can separate the adipocytes and FAPs into subtypes. We can look at the myeloid cells. They all have very, very clear and distinct markers. And the same is true for T cells, subtypes of T cells, NK cells, B lymphocytes, and other leukocytes. So we have a really, I think, high quality atlas of human adipose tissue here. And there's also actually quite good correspondence between the labels that we have derived and the original labels by the authors, even though, of course, the nomenclature is very different. So, for example, if you look at our adipocytes, and in the Rosen data, they have labeled them also adipocytes, and it's very, very, very clean. The same is true here for this. For both the data set, it's also very clean. So we did some analysis on this data set, but we actually mostly generated it to showcase what you can do with jointly. But we did a little bit. So particularly, we looked at compositional changes, of course, in obesity. And we saw some interesting things. So we see here that that to the left here means higher in obese individuals, and to the right means higher in lean individuals. We see some expected things. We see, for example, that there's an increase in the number of or in the proportion of macrophages in obesity. And we see, if we then zoom in on the subtypes of macrophages, that it is actually specifically the LPL positive or TREM2 positive macrophages, especially, that is driving this, which is consistent with literature. So on, we can see for different resolutions of cell types, we can see how they're changing across tissues or across obesity. We can do the same if we look between depots. So here to the left, we have the visceral adipose tissue, and to the right, we have the subcutaneous adipose tissue. And we can see, as we would expect, that mesothelial cells are only present in the visceral adipose tissue, and other sort of changes. So, for example, we see, interestingly, that FAP cells specifically is high in subcutaneous adipose tissue. So, just to round off the take-home messages, I think that I hope that I've convinced you that jointly can achieve state-of-the-art performance in batch integration tasks, and that it is also robust against overcorrection of heterogeneous samples. That could be multi-conditional, multi-sample conditions, such as having a cohort of healthy and sick individuals. And I think that the interpretable factors in jointly can provide the analysts with additional biological insight. Finally, I would say that jointly is very available. We have made both an R package that interfaces with RAD. We have also made a Python package that interfaces with ScanPy. And we have made all of the models and model weights for the atlas freely available to the community, so you can browse the data. You can also download the model weights to perform transfer learning of your own data sets or extend our atlas, if you will. The links for that is down here. So the final slide is just thanking the people who actually did it. So Andreas, who actually defended his PhD thesis last week, performed this work. He will join us very soon as a postdoc in HDRM. And I would just like to thank the other members of the group, of course, the Novo Nordisk Foundation Center and funders, and I'll be happy to take any questions.
Thank you, Jesper. That was awesome. Super, super interesting. Does anybody have a question?
I have kind of a philosophical question in general. We presented this as a batch correction method, and I think about batch correction like something in the past, like I don't know, combat that found the weights, and then you pass to the linear regression, and those will be regressed out. In your method, you're not really removing batches, but you are better finding a common and strong signal. Is that a fair assumption? Because I will never be able to know if the variability was for sequencing depth or it was for the assay that I've used, or if it was donor variability or sex that have been removed, right? There's no way for me to tell.
That's correct. So we do a few different things to focus on shared variability. One of the things that we have done is that we have implemented a consensus PCA approach, where we actually calculate the average covariance matrices and use that to perform the initial dimensional reduction, which focuses on shared variation between data sets. So the method is not removing any batch effects, but we are trying to reduce the impact on the latent space.
Yeah, makes sense. And another thing that I want to ask is, when you have you said that when you have multiple data sets, you run it independently and then you put everything together. Like and so how do you, is the number of factors always the same for each? I'm mentioning is the adipose example that you showed, where you said that you run everything independently and then you merge together. So the number of factors that are found for each of these data sets might be different, right? And then,
yes.
Then what you do, you correlate to find which one are actually the same. Like, how do you reintegrate given these matrices of factors that are individual for each data set?
Yeah, so in the VATLAS, we actually only use jointly to generate labels, and then we just supply those to supervised integration.
Okay.
So we don't actually use the latent space from jointly in that for other purposes than obtaining labels. We could conceivably have run this entire data set with a jointly. There's a few reasons that we didn't. One is that we wouldn't be able to extend it. And the other is that jointly being an NMF-based approach doesn't scale to millions of cells. Because the computations right now are done with dense matrices, and that is an area that we are working on, trying to implement, for example, sketching to improve the scalability of the method.
One last follow-up, and then I'll shut up, sorry. But if you're doing this way, the fact that you find the Schwann cells, so that means that already one data set had it. Like, this is not the power of, it happened a rare cell type or a hard cell type to sequence. You find only, I don't know, 10 cells for each of the data set. But when you put everything together, they become 100 and they are suddenly visible, right? Because you are integrating all the cells together and they have a strong signal. But in your case, basically is the fact that you are able to find that signal from the individual data set already had it and you were able to find it. It doesn't come from the power of integrating those cells together. Is that correct?
That's correct.
Okay.
Yeah.
Thank you.
Thank you.
I think in the interest of time, we should wrap up. Thanks again, Jesper.